Good morning students. Today we are going to continue our topic, that is the structure and function of the DNA. So you know that DNA or deoxyribonucleic acid and RNA, that is ribonucleic acid, these are the two major type of nucleic acid found in the cell. So yesterday we are talking about the DNA, that is deoxyribonucleic acid, and its structure. So yesterday we have already seen the function of the DNA. Today we are going to see in detail the structure of the DNA. So DNA is a double helical model, and a double helix has two strands twisted together. So here you can find out the two strands. This is the one strand and this is the another strand, green strand, which are twisted together. So DNA is a double helix and has two strands which are twisted together. And the one strand of the DNA, you know that DNA, it is consisting of the nucleotide, and nucleotide is consisting of the nucleoside. So what is nucleoside? So you can say that nucleotide, it is consisting of phosphate plus nitrogen base plus sugar. So when all these three content comes together, it forms the nucleotide. So DNA is consisting of small segments which carries the information that is said to be genes. Genes is nothing but it is the many units of nucleotides comes together to form the DNA. So you can say that DNA is consisting of nucleotide, and nucleotide is nothing but it is the addition of, or you can say it is the assembly of pentose sugar, nitrogen base, and the pentose sugar, nitrogen bases, and the phosphate. And this phosphate sugar and nitrogen bases, this come together and it forms the nucleoside. So nucleoside is nothing but it is combination of the phosphate sugar as well as the nitrogen base. So one can say that nucleoside is nothing but the combination of phosphate sugar and nitrogen bases. Here in this slide, one strand of the DNA, how does it look like? I said that DNA is consisting of nucleotide. So here you can find out the different nucleotides. So these are all the nucleotides. When these all nucleotides comes together, it forms the DNA. This is only the one strand of the DNA. So the one strand of the DNA is a polymer of nucleotides, and one strand of DNA has many millions of nucleotides. So you can find out this is one strand of the DNA and it has many nucleotides. And the nucleotides, you know, they are the structural unit of nucleic acid. So nucleotides are the structural unit of nucleic acids. Next, one strand of the DNA, you know that the backbone of the molecule is alternating phosphate and the deoxyribose sugar. So the backbone is consisting of the phosphate as well as the sugar. So this is the phosphate, and where is the sugar? This is the sugar. And there will be the nitrogen bases. So teeth like projection, what I can see, this is the nitrogen bases. The teeth are the nitrogen bases. So you can say that nucleotide is consisting of the phosphate plus sugar and the nitrogen bases. Nucleotides. So one deoxyribose together with its phosphate and base makes a nucleotide. I already said that phosphate, these are the triphosphate, that's why it is written 3 times. ATP, adenine triphosphate. So this ATP, this is a phosphate group, this is a sugar group. So this is in case of the RNA, but in case of the DNA, here the sugar will be the deoxyribose. So here deoxyribose sugar, which is a pentose sugar, attaches to the phosphate group, and this sugar is attached to the nitrogen base at the one end, and another base fifth position of the carbon with the phosphate. So in this way it forms a strand, one strand of the DNA, and in this way similarly the next strand of the DNA is formed. So nucleotides is a basic building block of the DNA and the RNA. So either it is consisting of, or it may be RNA or DNA. So what is the basic building block? That is nothing but that is nucleotide. And the DNA sugar is deoxyribose sugar, and the bases will be ATCG in case of DNA. But in case of RNA, the sugar will be the ribose. The sugar of the RNA will be ribose, and the basis will be A remains the same. Instead of T, U is present in case of the RNA. So just by seeing the bases, one can say that the information or the nucleotide may be of the RNA or the DNA. So in case of the nucleotide of the DNA, the sugar will be deoxyribose and the bases will be AT, C and G. A for adenine, T for thymine, C for cytosine and G for guanine. And in case of RNA, the sugar is ribose, and in case of the bases will be A, U, C, G. All the three bases remains the same. Only one basis, that is thymine, is replaced by means of the uracil. Uracil. Here we can see the sugar of both the deoxyribose DNA as well the sugar of the RNA. The main basic difference between these sugar, normally it is a pentose sugar, it starts with the, so pentose sugar, so 5 carbon atoms. So here this will be the first position, that is it is occupied by the oxygen. This is the first position of carbon, this is the second position of carbon, this is the third position, this is the fourth position of carbon, and one more carbon is present which is over here. So this is the fifth position of carbon. So here in case of the second position of carbon, here we can find out there is a presence of only one hydrogen atom over here. So this is why this is said to be the 2-deoxyribose, means here one oxygen is not there as compared to that of the ribose. Here in same case, if you see in the ribose over here, you can find out that second position is occupied of carbon is attached with the hydroxyl group. So OH group. So here if you replace it with the only H, then it is nothing but the deoxyribose, and if you replace it with the OH group, then it will be the ribose group. So this is the main difference between the deoxyribose sugar of the DNA and the ribose sugar of the RNA. So here two position is occupied in case of the DNA by only hydrogen atom, and here the two position is occupied by means of the OH group. Rest of the things will be remains similar. So here it is again consisting of 5 carbon atoms, 1, 2, 3, 4 and the fifth one. We know that the nucleotide is consisting of the phosphate plus sugar. Just now we have seen the structure of the sugar. Now is the time for nitrogen bases. So normally the nitrogen bases in the DNA there are 4 different bases, nitrogen bases, that is cytosine, thymine, adenine and the guanine. And we can say that These nitrogen bases again divide into two. They are the purines. They are further divided into two groups, that is purines and the pyrimidines. Pyrimidines. So nitrogen bases are grouped into two. They may be purines and the pyrimidines. And adenine and guanine, these purines, they are double ring. They are double ring compound. Whereas this cytosine and thiamine, they are the single ring compound. So you can say that cytosine and thiamine, they are the single ring compound, and adenine and guanine, they are the double ring compound. And that's why this adenine, which is a double ring compound, they are said to be the purines. They are the purines, and these are said to be the pyrimidines. So we can say that DNA has 4 nitrogen bases, either maybe 2 may be purines and 2 may be the pyrimidines. So what else will be the purine? The purine may be adenine or guanine, and the pyrimidines may be cytosine and the thiamine. And in case of the RNA, uracil, you can say that the uracil, which is again a single thiamine. Here thiamine is replaced by uracil. So uracil is again a pyrimidine, which is a single carbon, or you can say that they are the single ring compound. So nucleotides are named according to their nitrogen bases, and these nucleotides may be containing the thiamine. If the nucleotide they are containing the thiamine, it is said to be the thiamine nucleotide, and so on. And if it contains the adenine, then adenine nucleotide, thiamine nucleotide, cytosine nucleotide, uracil nucleotide. So the nucleoside refers to the combination of pentose sugar and a purine or a pyrimidine. So if a pentose sugar and either maybe purine or pyrimidine, any type of nitrogen bases will come together, then it will consisting of the nucleoside. It consists of the nucleoside. So we can say that nucleotide is again consisting of the nucleoside. It is observed that the nitrogen bases present on one strand pairs with, consider this is a one strand here, if these are the 3 different type of nitrogen bases which is present on one strand. So it is said that this A always pairs with the T, T always pairs with the A, and C always pairs with the G. So it is observed that the nitrogen bases which is present on one strand pairs with the nitrogen bases of opposite strand, and purine base A always pairs with the T. That is, you know that T is a pyrimidine base. So purine base A is a purine base. So purine base is always pairs with the pyrimidine bases. So A always pairs with the T, and C always pairs with the G. So you know that the purine G base always pairs with the pyrimidine C bases. So we can find out the ratio, that is 1:1. So purines, we can find out the purines is to pyrimidines. Or we can say that A pairs with the T, or T pairs with the A, and G pairs with the C, or C pairs with the G, with the 3 carbon compound. So what is the Chargaff rule? in the DNA molecules, total number of purine base is always equal to the total number of pyrimidine bases, and this is called the Chargaff rule. So what does the Chargaff rule says that total number of purines base is always equals to that of the pyrimidine, total number of the pyrimidine bases, and this is said to be the Chargaff's rule, and it gives the ratio. What is the ratio? Purines to the pyrimidine is always 1:1 ratio, and it can be represented as A plus G is equal to T plus C. So Chargaff rule, whenever you are asked, then it is nothing but the A plus G is equal to T plus C, or you can say that A plus G divided by T plus C is equals to 1. So this is said to be the Chargaff rule. For every purine present on one strand, there is a pyrimidines on the other strands, or the vice versa. So this is said to be the Chargaff's rule. Now coming up to the components of the DNA. So the DNA is that it is a polymer, or you can say that it is a biopolymer because it is present inside the organism, and the monomer units is of DNA. उसको DNA का एक जो पॉलीमर है उसका छोटा यूनिट को हम क्या बोलते हैं? We used to say the DNA. न्यूक्लिक एसिड का छोटा यूनिट को हम DNA बोलते हैं, and DNA is consisting of the nucleotides. So DNA is a polymer, and the monomer unit of DNA are nucleotides, and the polymer is known as polynucleotides. When many nucleotides will come together, they will form a polynucleotides. And each nucleotide consisting of 5 carbon sugar, a nitrogen containing bases attached to the sugar and the phosphate group. And we know that each nucleotide is consisting of sugar plus phosphate plus bases, nitrogen bases. And these are the 4 different type of nucleotide which is found in the DNA. We know it. And they differing only in the nitrogen bases. The 4 nucleotides are given 1 letter abbreviation as a shorthand for the 4 bases. So A stands for adenine, G stands for guanine, C for cytosine, and T stands for thymine. These are the 4 bases we have just now seen. So 2 kinds of bases in the DNA. So you know that it is consists of purines and pyrimidines. The purines, structure of the purines and pyrimidines is given over here. So I said that purines, it is a double ring compound. So you can find out the purines, which is a double ring compound over here. This is a single ring and this is another ring. So you can say that it is a double ring compound, and pyrimidines, they are the single ring compound or the bases. So pyrimidines and pyrimidines, these are the structure of the bases of purines as well as pyrimidines. So cytosine and thymine, they are said to be the pyrimidines, and pyrimidines, always remember, they are only consisting of only one ring. So here you can find out only one ring is present, and here it bonds with the another compound. So here you can find out the cytosine and thymine as a pyrimidines, and each one have a ring, one ring only of carbon and nitrogen atom. So these are the carbon rings, and here the nitrogen bases attached. And adenine and guanine, they are said to be the purines because they are the second or double ring compound. They are the double ring compound. They are consisting of two rings. So adenine and guanine each have 2 rings of carbon. So you can find out this is a 1 ring, this is another ring. So purine is a 2 ring compound of the carbon and the nitrogen atom. Now 2 strands of the DNA. So remember, DNA has 2 strands that fit together, something like a zipper. So here these are the two strands of the DNA, and here bases, teeth are the nitrogen. Here these are the bases, and these are the sugar and a phosphate. This is sugar, this is phosphate, and these are the nitrogen bases. And the teeth are the nitrogen bases, but why do they stick together? They attach क्या होता है? Because they contain the hydrogen bond. So the base attract each other because of the hydrogen bond, and the hydrogen bond are weak, but there are million and millions of them in a single molecules of the DNA. So because of its abundance, this hydrogen attraction takes place, and the hydrogen bond between the cytosine and the guanine, they are shown here with the dotted lines. So here you can find out the here dotted lines. These are nothing but the hydrogen bonding. So it is said that the bond formed between the cytosine and the guanine. So we know that cytosine is a pyrimidine, and it is consisting of only one cyclic ring, and guanine is a purine base, and that's why it is consisting of two cyclic rings. So here we can find out the guanine, and this is the adenine. So this is the cytosine, which is a single ring carbon compound, and this is the double ring carbon compound. This is purine. So this is pyrimidines and this is purines. So when purines and pyrimidines, they comes together with the means of hydrogen bond, then normally the dotted lines can be seen by means of the hydrogen bond. So hydrogen bond, when making hydrogen bond, cytosine always pairs up with the guanine, and adenine always pairs up with the thymine, and adenine is bonded to thymine here. So here we can say that adenine is based with the thymine, and here adenine and thymine is given with the two hydrogen bond. So today we stop here. Tomorrow we are going to continue the topic, and very soon we are going to finish this topic. Thank you for watching.